So for now, we start with the second talk in the session by Andrea Thorn. Andrea Thorn is a group leader at the Rudolf Virchow Center in Würzburg, a Center for Integrative and Translational Bioimaging. And I'm looking forward to your talk.
Thank you very much for having me here. I'm going to talk about the Coronavirus Structural Task Force inside Corona. So here on the title slide, you can see a picture of the virus, the bilipid membrane, the spikes, and all of these things that you see here are actually molecules. Now, the virus as it is usually depicted only shows the transport form, the virus particle. But once a host cell is infected, the virus produces a lot more molecules, in total about 29 different types of biological macromolecules, most of them proteins. They form the hull, the spikes, the replication machinery that makes more viruses. Here you can see a depiction of the main protease. You can also see a potential drug bound to this molecule, which would inhibit its function. Here is another picture of the spike. So in order to, as Frank just lined out, rationally design drugs, we need to know these structures. And that is what we as structural biologists usually do. We use 3 different methods: X-ray crystallography, nuclear magnetic resonance, and cryo-electron microscopy to elucidate molecular structures. In this case, the structures of SARS-CoV and SARS-CoV-2, which are very similar. SARS-CoV-2 is the current Covid-19 causing virus. But there's a problem with this, and that is our workflow. So first we biologically prepare the protein or the macromolecule and purify it. Then no matter what's the method, NMR, cryo-EM, crystallography, we do a data measurement. And then this data measurement is interpreted with a structural model in the computer. So the model that you see, the balls and sticks, are not the direct outcome of the measurement. They are merely a model that is interpreting these data. And the fit is usually not so great. If we go by R values in crystallography, a typical fit would be about 75%. So if the model resembles the data to 75%, we think that's good enough for publication and interpretation. And very often this is true, and it gives answers to our biological questions. But there are also downstream methods like in silico drug screening, any kind of drug discovery, bioinformatics, chemoinformatics, and of course, machine learning applications. And even with like biological questions alone, we sometimes get ligands like this one. If you are a chemist, you may notice that the geometry is not quite beautiful. And sometimes we publish structures and they get on the cover of Science, but because the model wasn't right, the article has to be retracted. So a structure can even be completely wrong. And that is just a matter of fact in our world, that structural biology does not always produce exactly right structures, but there are errors in it. And this happens even in the best labs. So what can we do? Now, add to these problems the following, right? As scientists who are working on coronavirus, we currently get a lot of pressure to be super fast in discovering a cure. This exacerbates the problem. And while the structural biology has swung into action very quickly, we're very efficiently dealing with this. Everyone's working together, it's great. We still may produce errors. And these errors may hinder downstream finding a drug. So as early as in February, I felt we needed to do something about this. And I started to look into structures, and I've quickly found colleagues like Tristan Kroll, who looked into RNA polymerase, who did the same thing. And this is just an example. RNA polymerase has been known from SARS, from SARS-CoV-2. The structure is a high impact, important structure. The RNA polymerase makes more genes. So if the RNA polymerase cannot make more copies of the RNA, new viruses can't have new RNA and no new viruses can be produced. So stopping this molecule would mean having a potential cure. However, the structure that had been deposited, and actually every RNA polymerase structure ever since the first one, so 7 or 8 structures, had the same problem with Tristan Kroll illustrates in this video of his software Isolde. There was a 9 residue out of register shift. Oh, it does not play. That's too unfortunate. And you would usually see here the shift of 9 residues, so the whole molecule changes its conformation as you see the error. This structure also contains a number of wrong magnesium ions and free diisophosphate that we think there's no experimental evidence for. And adenosine had been modeled backwards. There were 2 prolines which were ought to be cis and a few waters missing. So we added all of these and offered the structure back to the original authors as a corrected structure to deposit. And they luckily, I think, reversioned their file. So this is what we do. And we quickly became a bigger team. We called ourselves the Coronavirus Structural Task Force because we looked at those 500 known structures from coronavirus. These are some of our members. We're by today 15 people from 7 different nations. All of us are methods developers in structural biology, meaning we're specialists in solving structures. Usually we develop the software that people use to solve their structures. We aim to validate the existing structures and the new ones that come up every Wednesday, improve them manually where possible by re-elucidating the structure solution process, looking at the models and comparing them to the experimental data, then disseminate these models back to the original authors and to the scientific public in a GitHub repository. And we also try to put the structures into context for people who are new to coronavirus. For example, drug developers that were focusing on HIV and are now coming into the coronavirus field. We do this on a homepage, insidecorona.net. We also have some information there for the general public about how all of this works. And we have a repository and database of our validation results and structures, plus a small SQL database for the metadata, which can be found on GitHub. We also use, like many scientists now, Twitter. And if you're really bored, you can also watch us on Twitch while we work on structures. One thing that I wanted to quickly mention, one of the things that we do for validation is the software HARSPEX, which is a neural network and can interpret reconstruction maps from cryo-EM, like this one from the spike head, by secondary structure. So it can differentiate the reconstruction maps even before you have a model into what is helical and what is beta sheet. And I'm rather proud of this because the paper only came out like last week. So I wanted to quickly show this to you because also ELLIS is all about machine learning. And we're using this now as well to experimentally determine our structures. This is the homepage. Important sections are news, where you can find blog entries that line out different important topics, and for users, which is a section for downstream users. So this is the news section. We put online about 3 articles per week. We also invite guest contributions. And for users, it's a bit more technically and helps people to decide which of the 500 structures is the right one that they want to use for their question. We have a lot of downstream users, individual labs who email us sometimes. Sometimes we don't know about them. Sometimes we only learn about them when they have a problem and they contact us. Folding at Home uses our structures as the basis for their 2.4 exaflops, 1 million PC computer power. MolSSI is a simulation and drug discovery homepage that directly binds into our repository to pull the data. 3D BioNotes, and of course the Joint European Disruptive Initiative has started a big challenge where labs can win money for discovering the best drug. And these groups are also encouraged to use our data as a starting point. Of course, we're very happy to hear from you, just in case you want to use our data as well. And we're very happy to guide new users on how exactly to use the repository and try to answer their questions as far as we can. Now, what's the outlook of this? We would like more automatic validation and graphics. We still haven't, I think we're not at capacity to what we could do in terms of automatic validation. However, we are at capacity with personnel right now. We are looking to have a better interface from the homepage to the results. So a 3D view of the molecules, for example, would be nice. We are writing to individual structural authors for the raw data because that permits us to have the whole structure start to finish validated. And it's really valuable for us. It's a big argument that images should be deposited, but it's not universally standard in our community. So we're pushing for that, and we write to authors individually, and we also offer to deposit raw data for them. We also would like to have more insight into the virology of this virus. So we still would like to know more experts on coronavirus, because we are structural biology methods developers. And I think we would extremely profit in our oversight of the whole process, and also in our blog posts, if we could give more context. Future plans also include community-driven animation together with the IWASA lab. So we hope to make an animation of, for example, the virus entry, where then scientists can annotate inside the video, inside the animation, and say, right, this bit isn't right, look at that paper. And we termed this using internet rage for annotating things. And we would still like more context for structures. Now, at the moment, I'm also looking for student helpers for this project and a PhD candidate. So if you're interested in joining us, I can only hire student helpers from Germany at the moment due to regulations, but PhD students from all over the world, please contact us.